Hello everyone, this is the start of the Sophion hosted webinar on automated patch clamping and iPSC, a second part because we had another one just a few weeks ago by Mike Henderson of Brainzel and Daniel Sauter of Sophion. We've got another great lineup today. Before we actually go into it, I will just play a video for you because we're still waiting for a number of you to turn up. Thanks to you that turned up. You get the pleasure of a nice little video clip of our virtual lab tour, and then we'll just give the others a few more minutes to log in because we're expecting probably, I don't know, another 30 or 40 people maybe sort of turning up yet. So we'll just run a quick video for you just to let you know things are happening. We just want to wait for some of those slightly later people to log in. So if we can run that video, it'd be great. And I'm not sure if that video will run again, but we've got a few more people logged in yet. We're still probably waiting on a good 20 to 30 to log in, hopefully, based on the people who've accepted the invite. I don't know whether it's worth running it again, Jane, or is it good just to sort of for me to... I'll shut up. Okay, everyone. I've just noticed a message actually in the chat box that I think Ralph Kettenhofen may be having a problem with the login to the webinar. So hopefully he can get in. I think Jane's just responding now. But we've now got sort of a few of you logged on. I think there still may be a few more, but I will start the introductions because we're a few minutes after 5 now. We've got 2 really nice speakers for you today, talking about the recent advances that we've been making with a nice collaboration with Dr. Liz Buttermore at the Human Neuron Core at Boston Children's Hospital. So Liz will be introducing some of this iPSC work, basically how they're developing iPSCs in their labs, and the huge number of advances that seem to have been happening in the last couple of years at least, certainly in their lab, and we've been lucky enough to do some collaborating work with them upon. So whilst Liz will be introducing some of, I think, the molecular sort of biology around iPSC development and culturing, Cadler will then go on to talk about, my colleague in Sophion will go on to talk about our collaboration with Liz's team at the Boston Children's Hospital, talking about essentially automated patch clamp or APC characterization of the electrophysiology of these iPSC-derived neurons that Liz's group have been doing such a great job developing. I will give it a minute or 2 more, and what I will do is I'll actually sort of tell you about how this will proceed. So Liz will go first, followed by Cadler. We've got some questions under the polls tab. So on the right side of your screen, you'll hopefully see a chat, polls, and handouts sort of tabs. So there's a couple of questions, or 3 questions, sorry, in the polls. And we've got a number of PDFs that you can download in the handouts, which are application reports and posters relating to a lot of this iPSC work that some of you may find interesting and useful to sort of download and digest in your own time. If you can place your questions in the chat, we will have both talks, and then hopefully we'll have at least 10-15 minutes of Q&A for both Cadler and Liz to follow the end of both talks. So yeah, put any questions you have in the chat and we'll do our best to address them as best we can. If there are any that are left unanswered, we'll try and make sure that we send them through to the relevant speaker so that they can hopefully answer your question after the fact. Without further ado, I'll let Liz take over, and looking forward to some really nice talks. Over to you, Liz.
Thanks, Damien, for the introduction, and thanks for inviting me today to be a part of this webinar and tell you all about the work that we're doing at the Human Neuron Core at Boston Children's Hospital, and also with our collaboration with Sophion. So first, I wanted to start today by just taking a step back and looking at why are we using iPSC-derived neurons for drug discovery in neuroscience, and that takes first a look at what are the challenges that are facing drug discovery in neuroscience, and obviously the first is access, and that's always going to be a problem of overcoming the blood-brain barrier. The other is one of the reasons I got into neuroscience is that the biology of the brain and the nervous system is very complex, and the circuitry is really still being defined. Because of some of these difficulties, there's been few validated targets in the neuroscience space. And as you can see from the list that I provided here, many of these targets are decades old, ineffective, or have unwanted side effects. On the translatable side, there's also been a lack of translatable biomarkers moving from preclinical space into clinical trials. In clinical trials, there are difficulties for selecting the right patient population to have a successful clinical trial outcome. And so some of the considerations that are being discussed these days are thinking about whether certain populations that have defined genetic backgrounds or defined stages of disease should be included in trials, but we're still trying to figure out what those are. We don't know at what point in a neurodevelopmental or a neurodegenerative disease they can no longer be treated. But that might be important in stratifying success in a clinical trial. And there's also been a lot of variability in clinical endpoints to show a therapeutic benefit. In the neurosciences, a lot of these clinical endpoints may be subjective. And so thinking about how can we make better clinical endpoints that are more reproducible and robust. But for our part in the Human Neuron Core, what we're trying to do is improve identification of validated targets to try to move some of this work forward and improve drug discovery outcomes in neuroscience. So when we think about preclinical model systems in neuroscience, traditionally a lot of this work has been done using rodent models because they're easy to access the genetic background. We can make knockouts and understand how those knockouts change neuron development and neuron activity. But what we've really been seeing over the past few years is that these models are not translating well into the clinic. And this could be due in part to ineffective behavioral models from rodents, and that this rodent brain really can't recapitulate the complexity of the human brain. In addition, these rodent models do not reflect the genetic diversity of humans and are often inbred lines that are used for these studies. And while there has been a boom in human genetic data that can enhance our confidence in targets, it doesn't tell us when we should treat, how long to treat, or what the outcome measures will be that would demonstrate efficacy in these patient populations. Other model systems that have been used in preclinical neuroscience research are also heterologous expression systems or engineered cell lines where you can overexpress a particular ion channel of interest and screen for compounds that affect that ion channel. But these cell culture systems don't recapitulate the neuron signaling mechanisms downstream of those channels. So when we think about how can we improve our preclinical model systems, we want to think about improving human relevance, allowing for improved mechanistic insight, and high throughput. And this brings us to the crux of iPSC technology, or induced pluripotent stem cells, which will allow us to have improved both human relevance, throughput, and insight in the mechanism. So what are induced pluripotent stem cells, or stem cells? These are undifferentiated cells that have the ability to become any different type of cell in the body, and they have indefinite self-renewal. There are a few different sources of stem cells, but today I'm going to tell you about our work using induced pluripotent stem cells, which were discovered in 2006 by Shinya Yamanaka, where he found that 4 transcription factors can be expressed in terminally differentiated fibroblasts, and this makes them pluripotent again. The great thing about this technology is that now we have the ability to take a skin or blood sample from a patient and generate iPSCs and thus any cell type from these patients. So this really leads us to a lot of promise and exploring what the implications are for iPS research. So now we're able to take a somatic cell, a blood or skin cell from a patient, reprogram them into iPSCs, differentiate them into different subtypes of cells, neurons, or glia, and then create disease models in a dish. These disease models can help us understand molecular mechanisms of disease, but they can also be used for drug screening and drug discovery. These screens can also be phenotypic screens that help us, again, to understand the molecular mechanism of disease. These cells can also be used for toxicity tests to understand which drugs might be more toxic to neurons. And really together, these studies create the potential for what's being called human preclinical trials in a tube or in a dish. And so when I think about this, I like to think about the potential to create iPSC-derived neurons from an entire population of patients and see which ones are able to respond to certain therapeutic interventions and understand why on the molecular level. And this will really help us to identify new drugs that would then come back to the patients. So how do we create these human neurons without accessing this primary human tissue? There's a few different ways that we can differentiate stem cells into mature neurons for modeling. One of the ways is by using small molecules to mimic the developmental pathways that are seen during neurodevelopment. So you can see in the figure on the left how stem cells are differentiated into progenitors, and then exposure to different small molecules allows for either cortical or spinal cord subtypes of neurons to be differentiated. These protocols typically take multiple weeks, if not months, to create the final terminally differentiated cells, and often create very heterogeneous populations of cells. On the other hand, the figure on the right shows how in the recent years, transcription factors have been used not only to differentiate the patient fibroblasts into pluripotent stem cells, but then to take these pluripotent stem cells and directly differentiate them into the terminal neurons or oligodendrocytes or astrocytes or other glial cells with the ability to bypass this progenitor phase. These protocols are really useful because, for example, we can use neurogenin overexpression to create cortical neurons in 1 week instead of several months. So this allows for the process to be really sped up. However, if your project is interested in looking at the role of a particular patient variant in the process of development, this kind of differentiation with transcription factor overexpression will bypass those developmental stages and may not mimic that process exactly. But these populations are more homogeneous than the others, although there is still some mixture of neural subtypes in these differentiations. And they're great for screening because you can have more similarity across your cell culture. So when we think about on the big picture, what the strengths and limitations are of this technology, on one side, these cells are human, so there's a human relevance to the model system. They're scalable. We can obtain these from patient-relevant genetic backgrounds, and we can maintain the neurons in vitro for months, and some are showing maybe even years now. These cells are genetically modifiable, so we can now use CRISPR technology, for example, to correct a patient variant and see how that specific variant affects neuron biology. And we can create some well-defined cell types. And every day, these cell types and model systems are advancing, including now with the advances in 3D organoid differentiation, where we can create subsections of these organoids that look like cortical layers or inner ear organoids from other groups. The limitations of these this technology is that these neurons are still very immature for the most part, and they're more embryonic-like. This can be a struggle for groups that are trying to look at neurodegenerative diseases. These cells are also prone to karyotype changes, so the iPSCs need to be very carefully cultured. I think one of the biggest struggles in the field right now is that there's a lot of variability from line to line, clone to clone, differentiation to differentiation. And often groups that are publishing new protocols are doing this and optimizing them with their favorite iPSC line that behaves really well. And when you move to another iPSC line, there can be quite a bit of variability. So we're trying to understand how we can control that in the field. This technology is relatively expensive and labor intensive. And I should point out that the exact cell types we're producing, although they are somewhat well-defined, they're not precisely analogous to those in patients. So there can be changes in ion channels that you wouldn't maybe see normally together in a particular neuron subtype that we may be generating here with this technology. So with these kind of strengths and limitations in mind, at Boston Children's Hospital and the Translational Neuroscience Center, we established this pipeline where we have a group of researchers who are able to identify and consent patients to collect patient fibroblast or blood samples so that they can be differentiated, reprogrammed into iPSCs. We also use CRISPR technology to generate isogenic controls. Then we differentiate these neurons into patient-specific different subtypes of neurons. And we characterize them for expression of different markers, neuron morphology, subcompartment localization. In this picture, it's showing synapses or functional activity with the goal of identifying phenotypes that are different in the patient versus the control so that we can develop phenotypic screens to identify compounds that reverse those phenotypes. The Translational Neuroscience Center also has access to animal behavior facilities and facilities that can test the efficacy and safety of these compounds in animal models, and support for clinical trials where we also need work to be done to optimize natural history studies and biomarker identification so that when we identify a new drug, it's ready to be tested in these patient populations. And this pipeline has already shown some early success from others who have used human neurons to support the drug discovery process. And I just wanted to show you one example of this from a collaboration that was done between groups at Boston Children's Hospital, Harvard, and GSK. In this study, they used patient and familial ALS iPSC lines that were differentiated into motor neurons. As you may know, ALS is a devastating disorder that leads to loss of motor neurons and early death of these patients. This motor neuron from ALS patients showed increased firing, increased excitability, and increased cell death in vitro. And this group was able to find that retigabine, an FDA-approved drug for epilepsy, actually decreased this hyperexcitability in a dish and increased survival of the motor neurons. And this data was able to get retigabine approved for clinical trial work, which is now in a phase 2 clinical trial for patients with ALS. So working off this idea and these successes, we want to use this technology to fill the gap for rare neurodevelopmental disorders. So at Boston Children's, we have collected blood and fibroblast samples from over 610 patients and controls. We have iPSCs now from 119 patients and controls. And I just listed here a few of the disorders that we have collected these cells from that range from intellectual disability to seizure disorders, spinal muscular atrophy. So quite a range and not exhaustive here. We're always open for collaborators to work with the lines that we're collecting. But today, what I want to do is tell you about 2 case examples where we have used iPSC-derived neurons to model refractory epilepsy. And I want to do this in the context of 2 disorders, tuberous sclerosis complex, or TSC, and CDKL5 deficiency disorder, or CDD. So first, what is refractory epilepsy? It's characterized by a number of things, including persistent seizures that don't respond well to treatment, cognitive problems, mood disorders, neurochemical changes, but really for the patients, a restricted quality of life. And so this is why it's so important for us to come up with new therapeutics for patients with refractory epilepsy. And it's thought that refractory epilepsy is caused by an imbalance in excitatory and inhibitory inputs in the brain, although in each disorder, we want to have a better understanding of what are the molecular mechanisms leading to these imbalances. So TSC2 is caused by mutations in TSC1 or TSC2 genes. This leads to tumors in many parts of the body, including the brain. It's often detected in infancy or childhood and has many different symptoms, including seizures and cognitive disabilities, as I mentioned. There's also cortical tubers, which you can see here in this patient imaging, that is associated with seizures in patients who have TSC2 variants. So to study TSC2, first I want to show you where TSC1 and TSC2 reside in their functional pathways so that you can understand what the consequences of loss of function might be for these proteins. And I always love showing this figure because it's titled mTOR pathway at a glance. And it's actually a simplified version of the mTOR pathway. And so you can see how complex this signaling mechanisms are. There's a lot of feedback loops and a lot of mechanisms to regulate this pathway. And the downstream effectors of this pathway include things like autophagy, mitochondrial function, protein synthesis, and cell growth. So there's a lot of things that are affected in the cell when this pathway is disrupted. And you can see that TSC1 and TSC2 are right here in the heart of the pathway. So when you lose TSC2 function, it releases the brake on mTORC1 activity, and this results in increased mTORC1 activity. And so that'll be important later in the talk. But for us studying this in our core, we wanted to use a TSC2 allelic series. And what I mean by this is that we generated iPSCs from a patient who was heterozygous for a mutation in TSC2. We also collected a familial control who was had 2 healthy copies of TSC2. And then we took the patient line and used TALEN to introduce a variant to the healthy allele so that we have a hypomorphic or null TSC2 line. These 3 lines were then differentiated into cortical neurons using neurogenin 2 overexpression. So again, this forces the differentiation of the iPSCs to neurons within a week. And then we used a series of phenotypic assays to characterize these neurons. What we found in the TSC2 null neurons was that they had a significant increase in cell body size that you could actually see by eye in the culture. It's not often that you can look at a culture and actually see the phenotype by eye without the quantification. But this is showing you an example of how you can see that. And of course, we do have the quantification showing that there was actually an allelic dose response where loss of TSC2 increased cell body size as well as total neurite length, showing that mTOR activity is likely increased. We also ran whole transcriptome analysis of these iPSC-derived neurons and found some really interesting findings. So there were some pathways of genes that showed an allelic dose response. So here in the patient heterozygous on the left, the signal is somewhat decreased compared to the healthy control, but then the null line is even further decreased. But what was really also interesting to us was that there were subsets of genes where the genes weren't that different between the patient and control, but were significantly altered in the null line. So we wanted to look closer at this population of genes and why are they so misregulated when you lose 2 copies of functional TSC2. And what we found is that these genes that are affected in the biallelic loss, but not monoallelic loss of TSC2, were actually ones that are identified as change in cortical tubers. These include things like cell adhesion molecules, extracellular proteins, things of that sort. So it's possible that these cortical tubers have maybe second hits or are more similar to complete loss of TSC2 than the heterozygous. We also wanted to look at the functional readouts from these neurons and what are the functional consequences of loss of TSC2. So for this, we plated these cortical neurons onto multi-electrode plates. An example of a single well is here, where you can see there are 16 electrodes per well. The neurons sit on top of these electrodes, and the electrodes record the extracellular field potentials. On the right are a few example raster plots from a healthy control, the heterozygous patient, and the null TSC2 line. And in each row are representative spiking from a single electrode in a single well, and each dash is representative of a spike. So you can see in the TSC2 cells, there's quite a bit of hyperexcitability and increased firing, which I have quantified in the graph on the left here, compared to both the patient and control. And what was exciting for us is that we wanted to see how whether mTOR activity was responsible for this hyperexcitability. So we treated the cells with rapamycin, which is an mTOR inhibitor, and we were able to prevent the hyperexcitability. So you can see the dashed line here is the null line treated with rapamycin, and these lines did not develop hyperexcitability. And the reason this was so exciting for us was because years before this study, these mTOR inhibitors had been shown in rodent models of TSC2 to reduce seizure burden and seizure frequency. So in this graph on the right, the black lines are vehicle-treated rodents, and the gray lines are rapamycin-treated. And you can see how rapamycin prevents seizure frequency over time. Rapamycin has also been used in patient clinical trials. And here's an example showing how this treatment reduced seizure frequency in patients, both by parent report and EEG monitoring, showing that this is reducing seizure frequency in patients with TSC. However, the positive effects of rapamycin appear to decline after the second year of treatment. There's also a subset of patients who didn't respond to rapamycin. So this really tells us that there's still a need to identify more therapeutic options, new therapeutic options for reducing seizure frequency. And what our results have shown is that we can use these iPSC-derived neurons as this model system to identify these new treatments. So how can we do this? The first thing we can do is develop a phenotypic screen to look for compounds that reverse this hyperexcitability that we see in TSC iPSC-derived neurons. And hopefully that would help us understand other mechanisms or novel therapeutic targets that can reverse this hyperexcitability. I'll also bring you back to the idea of a clinical trial in a dish where we can obtain neurons from many patients who have refractory epilepsy and try to understand the diversity in the phenotypes across these patient populations and what treatments are able to reverse those phenotypes. And then again, with the goal of moving into translatable systems such as rodents to confirm these effects. So now I want to switch gears and tell you about how we can apply this system to another a model system, a disease in our group, a cohort in our group, that's called the CDKL5 deficiency disorder, or CDD. CDD is caused by loss of CDKL5 function. It results in early onset seizures, severe intellectual disability, motor delay, delayed speech, sleep problems, and GI problems, just to name a few. And really the only therapeutic options available right now are to try and treat these symptoms. So this patient group is in need of new therapeutic options. So to understand and try to treat CDD, we first have to ask, what does CDKL5 do? CDKL5 is a serine threonine kinase. A few of the putative targets of CDKL5 have only been started to be described in recent years, and they are present in many regions of the cell, including the cytoplasm, nucleus, and the plasma membrane. And you can see in this diagram that CDKL5 is localized in both the cytoplasm and can translocate to the nucleus based on its phosphorylation status. And depending on where it's located, it can regulate functions in microtubule development or gene expression, or the expression of ion channels in the plasma membrane. So you can imagine how this might be really important for neuron development, but we don't really understand what the specific consequences are. So some groups have started working with CDD, patient-derived neurons, to identify cellular phenotypes and disease mechanisms. And I just wanted to highlight a few of the more recent findings. So one group has used patient-derived neurons to show that neuroprogenitors actually proliferate more slowly in this patient population. So you can imagine how reduced proliferation of neuroprogenitors may slow neuron differentiation process, neurogenesis, during neurodevelopment in these patients. This group also saw an increase in phospho-S6, which is a readout of the mTOR pathway, and increased mTOR activity, which would suggest that there might be increased neurite outgrowth in these neurons. And that group did, in fact, find an increase in neurite outgrowth in the CDD patient-derived neurons compared to healthy controls. However, another group also published last year using CDKL5 patient-derived neurons and found a decrease in neurite outgrowth in these iPSC-derived neurons. And so we don't know whether these differences are due to differences in differentiation protocols used, culture conditions used, or maybe just differences in the patient lines that are used to identify these phenotypes. So one of our efforts to try to help answer these types of questions was to create a resource of patient-derived iPSCs with isogenic controls that could be available to the research community so that we can all be working on the same subsets of lines. So last year, we deposited 6 isogenic pairs with from patient lines with these variants in the CDKL5 gene and their isogenic controls, and they've been deposited at Coriell Institute so that researchers can have access to these important iPSC lines. For this project that we're talking about today, we utilized an iPSC line where a patient has a variant in the kinase domain of CDKL5 that is predicted to be a loss of function variant. We took this iPSC line and control and differentiated them into cortical neurons using a patterning-based approach that took 5 weeks to differentiate into early neurons. They were then cryopreserved and distributed to Sophion, as well as characterized internally in our group. So the first thing that we wanted to do was to characterize the neurons that we made from this differentiation of both CDD patient and control iPSCs. And so in this staining here, I'm showing Hook's nuclear stain, a TIJ1 neuronal stain, and a PAX6 neural progenitor stain. And what I hope you can appreciate in these representative images, in the merge especially, is that the control neurons were able to generate a nice healthy population of green TIJ1 positive neurons. There are still some neuroprogenitors here labeled in red by PAX6, which do eventually go on to also differentiate. However, when you look at the proband, there was a great increase in PAX6 red positive cells here, and it looks like, at least in this image, fewer TIJ1 positive cells. When we quantified this across many wells, we saw an increase in cell number in the proband cell line. This was not due to an increase in TIJ1 positive cells, but was due to an increase in PAX6 positive cells. So this tells us that there may be a delay in the differentiation in these patient-derived cells. We also ran a whole transcriptome analysis to try to understand these neurons better, and we found that there were pathways of disrupted expression of synaptic machinery, of kinases, and importantly, of sodium and potassium channels. And so this was really interesting to us to see whether the effects on these ion channels might have some consequence to the function of these CDD patient-derived neurons. And there has been a group who published some answer to this functional consequence of loss of CDKL5 function, where they placed their cortical organoids on an MEA plate and recorded activity over time. They found that there was a hyperexcitability of these neurons about midway through the development and recording, but that this went away over time. And this is a little bit confusing to us as in the field because these patients have pretty severe epilepsy. So we don't understand how they could be similar to controls later in development. But that's why this collaboration with Sophion is so important, and Kadla will tell you a little bit more about the characterization of how these patient control lines are functionally different. But before I turn it over to Kadla, I just want to thank the team at Boston Children's Hospital. So I work with a very talented group of people in the Human Neuron Core. We also have collaborated with the Sophion Lab for the TSC2 project, and the members of the Human Neuron Core specifically that worked on the CDKL5 project that have now moved on were Ivy Chen and Taylor Foreman. And of course, our funding sources, including funding for this project with support from Sophion, and the other funding sources that help establish the core and our important work. I look forward to your questions, and I'll hand it over to Kadla to tell you about the rest of this exciting project.
Thank you. Thank you, Liz. I will just share my presentation. I hope that you are all able to see it. Otherwise, I guess Jane will let me know. All right. Yeah, so I'm Kadla. I'm a research scientist at Sophion, and I was responsible for the electrophysiological characterization of these iPSC-derived cortical neurons using automated PET clamp. And these preliminary results that we obtained is what I will present to you today. So as Liz already described, this iPSC technology has now given access, or relatively easy access, to neurological disease models, but that has also created a necessity for high throughput methods for characterizing and screening these cells. And since ion channels are implicated in neurological disorders, for example, Alzheimer's, Parkinson's, ALS, and epilepsy that we will talk about today, we started this collaboration with Boston Children's Hospital to develop an automated PET clamp assay for characterization and screening of iPSC-derived cortical neurons. My talk here today will contain a little bit of assay development. Then I will talk about or give an example of a biophysical characterization of the voltage-gated sodium current that we measured in these cells. And finally, I will show some preliminary results from a screening we did of the iPSC cortical neurons derived from an epilepsy patient. So, as Liz already mentioned, the cells, the neurons are, or the iPSCs are differentiated for about 5 weeks in the laboratory of Boston Children's Hospital. At this point, they are cryopreserved and they were shipped to us, and then we thawed them and matured the cells for an additional 3 weeks before we did our measurements. So I refer to this time point when we thaw them as day 0 in vitro. One of the challenges of performing automated patch clamp on iPSC-derived cell lines is that we have to have the cells free in suspension, which means that we have to dissociate the cells. As you can see in the micrographs here, the neurons, they form a quite extensive and complex network, and it is hard to dissociate them without harming the cells. And what we really need is a cell suspension in which the cells are viable and in which there are limited debris and preferably no aggregates. So we have spent quite some time looking into different harvest procedures and protocols, and we have tested a lot of things. And different cell lines all have different requirements. But the top 3 here is what we used for this cell line, and there are many more things that you can do to make sure that you have a nice cell suspension. And once you have that, you can perform your experiment. And all the data I will show you today was performed on our high throughput system, the Qchip. For this, we used something called a cell transfer plate. It has these 4 compartments for testing different cell clones. We use 680 microliters per well, and we recommend a minimum cell density of 1.5 millions per milliliter. The consumable that we use on the Qchip is called a Qchip. It has 384 sites. And this I show over here is the Qchip plate view. With the cortical neurons, we reach up to 50% success rates, which is here color-coded green. And on each side, there will be a single cell where you can apply your voltage step and current, or voltage clamp and current clamp protocols as you desire, and measure the output. So I mentioned briefly the cell density. So for the cortical neurons, we actually performed experiments at many different densities. So here we plotted the density as a function of success rate. And as you can see, the data is quite scattered. But when we bin it, there is bin the data together in these data points, there is a trend, which is not surprising to us. This is something we see for all cell lines, that when you decrease the cell density, you also decrease the success rate. And we recommend 1.5 millions per milliliter for this type of cell lines, but you can of course go down further. We know that so iPSC cell lines are expensive, and sometimes you don't have so many of them. So you might need to go down in density further, but you risk then compromising the success rate. Then one of the applications that we believe, or where we believe APC is really or automated patch clamp is really promising in conjunction with iPSC-derived cell lines, is the ability to identify, well, both characterize cell populations, but also identify subpopulations within the bigger population based on their ion channel currents. So here you see a crop out of the Qchip plate view. and we have applied some quality filters on the membrane resistance and on the cell capacitance. And you can already see here now the green labeled sites have passed the filters, and the red labeled sites have failed the filters. So there are already some sites that have failed based on these quality filters, but then we can, based on our knowledge of the cells and the ion channels that we expect to find in the cells, we can add some additional filtering on those different ion channels. So in this case, we filtered on the voltage-gated sodium current, we filtered on the potassium current, and then we also looked specifically for A-type potassium currents. And then you can see this further filtering. Actually, there are some of the cells that don't contain all of the ion channels that we are looking for, so they are filtered out. And then you can identify the subpopulation of cells that you wish to work with and analyze that further. And what we used this for in this case was that we compared 3 different batches of cells. They were all differentiated from the same donor, but they were differentiated at 3 different time points. And often we see a large variation in the ion channel content of different batches of iPSC-derived cell lines, but here we were actually not able to detect any difference within error. So you can see there is quite some variation between the individual experiment, and within this error we did not see a significant difference between the 3 batches, which is a very good starting point. So this is in gray is the voltage-gated, the cells that were in which we detected voltage-gated sodium current. In darker gray, we have the voltage-gated potassium current, and out of these, the striped bar here is the A-type potassium currents. So then I just wanted to give you an example of a biophysical characterization of the voltage-gated sodium current that we did here. You can see here the current voltage relationship that was plotted for a control cell line in black before and in white after the addition of 1 micromolar TTX. And what we saw, as expected, was that all the voltage-gated sodium channels expressed were TTX sensitive. Then we went a bit more into details to try and to see if we could get some insights about which ion channel we primarily saw expressed in the cells. So we plotted the current normalized to the maximum current as a function of voltage to obtain the inactivation curve, and we calculated the conductance and normalized that to get the activation curve. And fitting then the Boltzmann equation allowed us to extract the V-half of activation and inactivation, which we saw aligned most closely with literature values for the NAV 1.2 channel. So this indicates that the majority of the neurons we are measuring are excitatory and that they are not mature enough to express high levels of NAV 1.6. And as Liz said, this immaturity is something that is observed quite often with iPSC-derived cell lines. It is a widely known problem that in general they are immature as compared to the primary counterparts. Then the last thing is the preliminary results of this screen that we did of the cortical neurons that were derived from a CDD patient. And Liz already introduced it. CDD stands for CDKL5 deficiency disorder. This protein has kinase activity, and it is present in the cytosol as well as the nucleus and involved in numerous cellular pathways. So the CDD is caused by mutations in this protein that results in decreased activity, or maybe a complete removal of the activity. That has, of course, a lot of consequences for the cell that eventually leads to a delay in neurodevelopment, autistic features, and early onset seizures, as is observed in these epilepsy patients. So we worked then with neurons that were derived from a patient that carried a missense mutation in CDKL5, and then we worked in parallel with the isogenic control, so a control cell line where this mutation had been corrected. So we had when we work on the Qchip, we use the cell transfer plate, as I mentioned already before, and we have here 4 different cell compartments, and we added in the top one the control cell line, and the bottom ones the disease cell line, and consequently the measurement plate laid out looked like this. So the top half of the plate was the experiment was run on control cells, and in the bottom part on the disease cell line. Then for the membrane parameters that we extract initially and can compare, we can see that the membrane resistance was similar for the 2 populations of cells. But what we saw was actually that there is a significant increase in the cell capacitance, and that is proportional to cell size. And together with the imaging data that Liz showed, this indicates quite strongly that the neurons are more immature than control neurons, and that the differentiation is not happening as efficiently. Then we can also extract the resting membrane potential. We did not detect a difference between the 2 populations, but I wanted to still show it because it shows a problem that we have with these cell lines, and that is that they often have a quite elevated resting membrane potential as compared to what we would have expected. And this can both be due to lacking ion channels and the immature phenotype of the cells. This can also be a consequence of us having to dissociate the cells, so they lose their morphology, and therefore their ion channel content might change. But this is at least something that we observe a lot, and that we are working to improve. Then we compared the voltage-gated potassium channel currents that we observed. So out here on the left is the current voltage relationship, and you see here the control in black before, and then in white after we add a block of 1 millimolar of 4-aminopyridine, and we see about 80% block. And then we observed on the steady state potassium current a small but significant decrease in the current in the disease cell line. And this still has to be validated that it's actually a significant difference, but at least it indicates that there is a difference, and that is in agreement with the sequencing data that showed that there was differences in the expression of several potassium channel subunits. Then more studies are needed in order to identify exactly which one and what it is that is happening. We did not see any difference in the peak current. So just to summarize a little bit, I showed you here an ion channel analysis of the cortical neurons that revealed that only a subpopulation of the neurons expressed the voltage-gated sodium and potassium channels that we were expecting. These expression levels were comparable within error between 3 different batches. And then we did a biophysical evaluation of the voltage-gated sodium currents that could identify that the neurons predominantly express the NAV 1.2 channel. Then we did screening of this CDD disease cell line, and the APC data together with imaging and sequencing data suggest that these disease cell neurons are larger and more immature than their control neurons, and that they might display differences in potassium channel expression. So other things that would be interesting to look into is, for example, to compare glutamate-induced currents, because we saw also from sequencing data, and it has also been published, that there is a upregulation of the NMDA subunit. And then, of course, we would like also to perform current clamp action potential measurements. So this is possible. We can measure action potentials from these cells, but the success rate is still limited for numerous reasons. And one of the strategies that we would like to pursue, or that we are pursuing to improve this, is to perform perforated patch clamp, which we can do on the Qchip. Yeah, then just to say that we believe that the APC in conjunction with iPSC-derived cell lines have great potential, both within the characterization of quality control, disease modeling, drug screening and pharmacology, and maybe even personalized medicine. And then finally, I wanted to mention that we have actually worked now with several different types of neurons. We have also collaborations where we work with cardiomyocytes, and we are starting also to develop assays for primary cell lines. And we are always open for collaborations, so if you have interesting cell lines that you think we should try out, contact us through the webpage. Also, if you're a master student or a PhD or early researcher, we have these travel and research grants that you can apply for. Yeah, that's it. Thank you for listening, and I will stop sharing.
I don't look like my video is. My audio was. Yeah, lovely talks both. That was really nice. And I think it's just a testament to the progress that you're making, Liz, obviously at the Human Neuron Core at Boston Children's Hospital, and also how we're trying to apply it on APC to actually, give high throughput phenotypic characterization via electrophysiological methods. We've actually got a number of questions, so I'm going to try and get through as many as we can. I'm hoping that both Liz and Cadler can stay on for as long as they possibly can, but obviously if you do need to go, then we will have to let you go, I guess.
No, it's fine.
Equally, the audience, if you can stick around, please do, because we've obviously got a few questions to get through. Let me try and get through some of these now. Good thing is there's quite a few, so it means that you're not going to get my stupid questions. So I'm going to go to the very first one because it seems only fair. So I think this is probably for Liz because this came quite early on. Sumin asks, did you detect differences in the number of synapses each cell makes between wild-type TSC2 knockdown knockouts?
That's a great question. So we did quantify synapse number with some high content imaging experiments, and we didn't see any significant differences in synapse number across the lines. We only looked at one time point after dissociation and neuron maturation. So it's possible that over time that would change, but at least for the time point that we looked at, we did not see a difference.
Thank you. And I think this is probably also for Liz. Well, it's actually for both in some ways. How much does the dissociation of the neuronal cultures influence the electrophysiological properties of the neurons? I assume the neurites are lost during dissociation.
That's a great question. So I can answer for our side. When we dissociate the neurons, it takes at least a week for baseline neuronal activity to start to recover. And then usually 2 or 3 weeks later, we feel like we're looking at typical neuron activity levels.
Yeah, I agree. What we see is that I don't think that the neurites are cut off. I think they are retracted during the process, so we end up with these ground cells. And of course, that really affects the distribution of the ion channels. So you need to really be careful what you're looking for and what you're looking at. When we can detect that the ion channels are still there, then you can characterize them. And also we can see that we can measure action potential still. But of course, anything that has to do with their morphology or with their communication, that is lost in this process.
Thanks. Anna asks, and again, this is pretty much to both of you, were these cells tested on the Qube also tested on MEA? Is the immaturity in terms of ion channel expression reflected in the firing activity?
Yeah, another good question. So we actually just received a grant to do that experiment. So we haven't done it yet, but we're excited to see how these look on the MEA as well.
Yeah, and in general, just to add to that, we are very interested to do comparison across platforms, comparisons with MEA, but also with manual patch clamp. So if anyone is interested in doing that, because this information is really crucial to understand what you can actually examine and what you cannot with this method. APC has the throughput, but it does not have the connections and all that. Yeah.
Thank you. Jean-Francois, hello, Jeff, how are you doing? Anyway, get on to Jean-Francois's question. You talked about the cell density, but what about the volume for each of the 4 compartments of the cell hotel or the cell sort of plate that we have?
Yeah, I think I noted it down that for this one, it's 680 microliters, but we actually have cell transfer plates with up to, I think, 32 tiny wells, and you can then it's just scaled down linearly the volume. So if you have very few cells, then you can actually use a plate like that, and then you fill up the rest with other types of cells. So that is an option, because I know with these cells sometimes you don't have so many.
And Kelly asks, and I think this is probably for Cadler, since it's relating to some of your data, how does the increase in cell capacitance relate to the maturity of the CDD cells?
Yeah, that's a good question. I'm actually not a neuron expert, but I think Liz and I discussed it, and Liz agreed that slightly larger cells is a sign of immaturity. Is that correct, Liz?
Yeah, I can expand on it a little bit. So during the differentiation process, we're going through these neuroprogenitor phases, and the neuroprogenitors are often have a larger cell body size, and they're bigger and flatter cells. And then as the neurons mature, they kind of round up, and the cell body becomes smaller. So it's possible that the increased capacitance is due to them being more immature. It is also possible that similar to the TSC2, if mTOR pathway is hyperactivated, maybe the cells are increased in size for that reason, but we haven't seen that by eye and in our initial quantification the same way as the TSC2 cells.
Thank you. And this is to you both. Have you used specific culture media like brain fizz that promotes rather than inhibits spontaneous neural activity, and so probably helps with the cell maturation?
That's a great question. We, in the human neuron core, have tested brain fizz before. I know it's used pretty widely in different culture systems. I think it's important when choosing media systems to think about how they affect the phenotype that you're looking at. So brain fizz may also increase hyperexcitability in cells, in some neuron cell lines. So we've chosen not to use it for some of these epilepsy model systems because we don't want to introduce ionic concentrations that are not physiologically relevant and that might alter the phenotypes that we're seeing.
And since I'm the host and I can question two experts who can't get away now, you've got to answer this. Kind of a follow on from David's question, actually, because obviously one of the challenges that you highlighted, Liz, was this lack of maturity. And clearly what you've been doing probably in literally the last 12 to 24 months now is you've been getting greater and greater capabilities in terms of the length of duration, which presumably is always adding to that ability to get more and more maturity in your culture growths. One of the sort of things that you're obviously aiming for ultimately is this, which I think is a really nice idea, this clinical trial in a dish. Do you think that clinical trial in a dish where you've essentially got a sufficiently robust cell line, a sufficiently robust assay upon that cell line, sufficiently mature cell lines, or you've made them sufficiently mature through your various developments in culturing and stuff, do you think that clinical trial in a dish is far away, as in, you know, you would at some point down the line, hopefully not too distant future, you'd almost have an FDA approved and certified cell line assay that you could be, as you say, use a clinical trial in a dish?
Yeah, I mean, I don't think it's that far off. Certainly in my career, I hope to see that, and not even, you know, early distance. I think it depends on what the indication is, what the need for the patient population and the risk for level versus what's available for treatment. So when you think about the ALS population, for example, they're a very high need population with no therapeutic options, who may be less risk averse because their deadline, you know, their timeline is ticking faster than a neurodevelopmental patient, which is a child who might be a little bit more risk averse in thinking about moving straight from a iPSC culture to the clinic. But it also depends on, you know, what kind of assay you're trying to build, or what the answer is. Are you trying to repurpose a drug that's already FDA approved, or are you trying to identify new mechanisms and new targets? And so those new targets, the new drugs, I think will still need the traditional drug development pathway, but the drug repurposing platforms, I hope, are, you know, closer to becoming more widespread in their translation to the clinic.
Thanks, Liz. Can I throw that to you as well, Cadla? Because I mean, you've obviously, you know, as well as Liz's great sort of work that she's been doing at Boston's Children's Hospital, you've been working on several different cell lines across different groups across the world. Do you kind of have a feeling, you know, it obviously doesn't necessarily need to be in the sort of neuronal area, although you've obviously sort of tested that too, but also cardiomyocytes is another big area that you've been working on. Do you have a feeling about that level of maturity and almost the robustness of some of the cells and the assays that are now being performed on these cells?
Yeah, I think there are still challenges, definitely, especially regarding the maturation. I think, but really just within the last 5 years, there has been a lot of improvement within, from what we see within the maturity markers that we are looking for. So I think it definitely will happen, but I think it has also been more challenging than expected, maybe, when we started out working with these cells. I think there's still some work to do regarding the quality control and the standardization of both preparing the iPSCs and also the differentiation methods. There are a lot of things that we still don't understand and that we have to control for, introducing mutations and all these things. I hope that APC could help some of it because it has the throughput that is necessary. But obviously, there's also things that we cannot do because we are dissociating the cells.
Yeah. Okay, well, I just want to thank you both for 2 excellent talks, and it's been a great collaboration, I know, from our side at Sophion with the human brain human neuron course, at Boston's Children's Hospital. So we want to sort of thank Dr. Buttermore and her team for all their great work. Thanks, Carla, for some beautiful data on the Cube and the Qpatch. With that, I would, like to sort of thank all the audience too for your time. It's great when we get so many people sort of logging in, and we hope these are useful. Of course, if you do have further questions arising afterwards, we'll be sure to push them through to Cadler and Liz. But thank you both, and thanks to everyone who's logged in. Take care. Goodbye.
Thank you. Bye.